Welcome to my presentation on lipids. So what are the main characteristics of lipids? There are three elements that all lipids have in common: carbon, hydrogen and oxygen. Lipids are insoluble in water, and therefore lipids will only be soluble in organic solvents such as alcohols. Examples of alcohols could be ethanol or propanol. Examples of lipids may include triglycerides and phospholipids. So what are some of the main roles of lipids? Lipids act as a source of energy due to the presence of a large number of CH bonds. This is because the CH bonds within the fatty acid chains store lots of energy and require lots of energy to break. Therefore, when they break, they release a lot of energy too. Lipids also act as a very important waterproofing material, as they are insoluble in water, therefore can act as a natural barrier. For example, in the waxy cuticles of plants, the waxy cuticle will help to reduce the rate of transpiration. Lipids can also be used as part of insulation, as they are slow conductors of heat, therefore helping to keep us warm. They also act as electrical insulators, for example in the myelin sheath of neurons. As a result, this helps to speed up the electrical activity. Lipids are also used to protect vital organs, such as the kidneys. Some key terms that we should know are that saturated means having no carbon-carbon double bonds. Monounsaturated means having one carbon-carbon double bond in the fatty acid chain. Polysaturated means having more than one carbon-carbon double bond in the fatty acid chain. Triglycerides are a type of lipid made up of 3 fatty acids and 1 glycerol molecule. Each fatty acid forms an ester bond with a glycerol molecule in a reaction known as a condensation reaction. Therefore, 3 molecules of water will be produced for in forming every triglyceride. Triglycerides store a lot of energy due to the presence of a large number of CH bonds. They have a low mass to energy ratio and can store lots of energy in small space, making them a very good energy storage molecule. They are large and non-polar, and thus do not affect the water potential of the cell. Upon oxidation, triglycerides will release a lot of water, which is an important source of water for animals living, especially in the desert. Phospholipids, on the other hand, are made up of a single glycerol molecule that is attached to 2 fatty acids and a phosphate molecule. The phospholipids are polar because their phosphate heads are hydrophilic, whereas their fatty acid tails are hydrophobic, thus allowing the formation of a lipid bilayer, for example in the cell membranes. This means that phospholipids allow the cell membrane to be selectively permeable about what they allow through. To test for lipids, we do something known as the emulsion test, whereby we add 2 centimeters cubed of the test sample to a test tube, followed by 5 centimeters cubed of ethanol, which we shake thoroughly to dissolve the lipid within the ethanol. Next, we would add 5 centimeters cubed of water to the test tube. The lipid will then precipitate out, as it is insoluble in water, and therefore will show up as a white precipitate, which indicates the presence of a lipid. Thank you for watching my presentation. If you liked it, please subscribe.